Well, thank you very much. It's my great pleasure to introduce the last speaker for this afternoon, Kristina. Oh, we've got two more, sorry. I'm very short-sighted, I apologize. The next speaker of the session, Kristina Neri. Kristina Neri represents the Systems Modeling Working Group. Kristina has had a very long-standing interest in the multidimensionality of different types of data and different types of species. So different cell types, different organs, time as a factor is just a few examples for some of these dimensions. Now, how to integrate this is a very challenging task in which Kristina has a keen interest. So, Kristina, thank you very much for speaking to us.
Thanks a lot, Michael, for the introduction and invitation to talk. Also to Kristina. So our system modeling working group is facilitated by Juan Botas, who couldn't make it. Apologies, and myself. I will explain what we do, but first I will explain who we are. So we are a multidisciplinary group of researchers bringing together clinicians, biologists, mathematicians, bioinformaticians, and basically what we do in the working group, we share views, we share data, and we provide advices on current questions, hot topics in HD research. And our focus, obviously, is to be data-driven for an unbiased understanding of HD molecular systems. There is this illustration to the right of the slide where you can appreciate the complexity of the data that we are dealing with, you know, across KEGG repeats, across age points, across cell types, and so on. So basically, in terms of the productivity of the working group, it's about basically the productivity of the member labs. And over the past years, new models were designed, new mixed data were generated, innovative computational methods were designed and used. We found new interesting targets, and we have a set of tools that are validated for the community to use on different topics. And you can basically access this information on the website of the working group. You can look at the publication list. These publications come from different labs in Europe, in France, in Germany, in the Netherlands, but also from the US. We have participants from several universities like UCLA, MIT, Harvard, and so on. CHDI is also a traditional member of our annual meetings, and there is really a huge level of expertise in this working group, and we mean to make it to enhance the utility of that expertise, to leverage this expertise for the purpose of the network. So the idea is that basically we need to understand the dynamics of HD molecular systems to better support target and biomarker discovery. And for example, the question that we are addressing are what are the cell type specific systems, you know, in neurons versus glia? What is the temporal dynamics of the HD process? Are we talking about linear process? Are we talking about critical periods? What are the links between disease modifiers and layer of molecular regulation, epigenetic regulation, transcriptomic regulation? What happens at the protein level? What happens at the level of intercellular communication? And we are very much interested, for example, in understanding the dynamics of pathogenic causative mechanisms versus compensatory response that may protect from HD. At the methodological level, we are basically giving a priority to building better in silico models that are increasingly useful for either target selection or biomarker discovery. In terms of what we are doing now, what we have been discussing during this working group meeting this year, after almost 2 years of interruption of activity or difficult activity of the working group, are about the production, analysis, and integration of data that are obtained in HD mice or in human HD samples. There is a lot of thinking today about collecting more information from human HD brains using the last technologies that are available today, like single nuclei analysis and so on. We are also planning for position papers and reviews on specific topics in the field of HD research. For example, what is the importance of causality or pathogenicity versus compensation? We are also working on developing new database for the community where people will be able to, in a very fast and easy, accessible manner, to get information on specific genes or pathways that are of interest, where they will be able, by entering these genes and pathway of interest, to know what they do across all the models of HD pathogenesis, you know, and so we are basically relying on omics, digital information, of course, which is an emerging aspect here, and AI. Now, just one example of the tools that we are developing, and I'd just like to take the example of what is being developed in my lab in Paris, where we developed the Biogemics platform, which is a precision machine learning platform to leverage complex omics data. So we have, as you can see here, a series of tools that we can use to analyze complex data, network analysis tools, shape analysis tools. There is a poster, 839, by Lucille Maigret, who is with us today. So if you can see this poster, I think you will understand what is shape analysis, computational statistics, deep learning, and also HD knowledge base. And I would like to invite you to look at the Brain Lab HD knowledge base, which is really a prototype for us to build a better database that will integrate a larger number of data, not only what we generated in our lab, but also what is being generated elsewhere and published by other labs. So if you want to learn more, sorry, if you want to learn more about the working group, who is there, if you want to contribute, just be in touch with us, and we will be very happy to discuss the needs and to discuss new projects. Thank you.
Thank you very much, Christian. Any questions for Christian? Yeah, Rosanna, we need a microphone.
In principle, how open is this group to integrate also data from other neurodegenerative diseases that might share some mechanism with Huntington? How open is this network, or what are the requisites to consider the possibility to include other neurodegenerative diseases?
Completely open. We have a task about HD research, but of course, I mean, to reach out to other neurodegenerative disease is very interesting. So because we can learn more. So yes, we are open to this, definitely. And if there are labs that want to contribute that kind of data or that kind of information, they are welcome to be in touch with us.